This presentation will cover what is CRISPR, the discovery and invention of CRISPR, definitions of Cas9 and gRNA. It will highlight how does CRISPR work, the types of genetic edits performed to use in CRISPR. It will also show how this technology is used for sickle cell disease and shed some light on ethical concerns with the use of CRISPR. So we'll start off by looking at what is CRISPR. So we'll start clustered regularly interspaced short palindromic repeats, or CRISPR, is a groundbreaking genome editing tool that allows for precise DNA modifications, with substantial applications in genetic research, medicine, and agriculture. The technique employs a guide RNA to target certain DNA sequences, which are then cut by the Cas19 enzyme, enabling gene editing. CRISPR is also utilized to construct genetically modified organisms for agricultural usage, allowing for the generation of pest, disease, and environmental stress-resistant crops. Furthermore, CRISPR has uses in biological research, such as studying gene functions, disease mechanisms, and developing new medications.
Discovery or invention of CRISPR. Discovery or invention of CRISPR. CRISPR, clustered regularly interspaced short palindromic repeats, was discovered in bacteria in the late 1980s and early 1990s by researchers studying bacterial genomes. Key milestones in its discovery and development include 1. 1987, Yoshizumi Ishino and colleagues first reported unusual DNA sequences, now known as CRISPR, in Escherichia coli. 2. 2001 to 2005, multiple researchers, including Francisco Mojica, Ruth Janssen, and others, identified CRISPR as a bacterial defense system against viruses. 3. 2005, Alexander Bulletin linked CRISPR sequences to Cas CRISPR-associated proteins, further clarifying its function. The invention of CRISPR as a genome editing tool is credited to Jennifer Doudna and Emmanuelle Charpentier. In 2012, they were described how the CRISPR-Cas9 system could be adapted for targeted genome editing in living organisms. Feng Zhang and colleagues in 2013, they successfully demonstrated the use of CRISPR-Cas9 in mammalian cells, advancing its application. What is Cas9 and gRNA? What is Cas9 and Cas9? CRISPR-associated protein 9 is an enzyme that plays an important role in the CRISPR-Cas9 gene editing system. Cas9 acts as a molecular scissors, cutting DNA strands, enabling medical scientists or researchers to edit parts of genome by adding, removing, or altering DNA within a living organism's genome. gRNA, or guide RNA, is a small RNA molecule designed to direct the Cas9 enzyme to a specific location on a DNA molecule. It is composed of two parts. 1. CRISPR RNA. 2. Transactivating CRISPR RNA. How does CRISPR work? How does CRISPR work? CRISPR is a powerful and precise tool used to edit genes within living organisms. It uses a protein called Cas9 and a guide RNA to target and cut specific DNA sequences. The guide RNA directs Cas9 to the right location in the genome, where Cas9 makes a precise cut. The cell then repairs the DNA, either by introducing mutations, disrupting a gene, or by using a provided template to make specific edits. This allows medical scientists to add, remove, or modify genes in living organisms with high precision.
Types of genetic edits performed using CRISPR. Types of genetic edits that can be performed using CRISPR include genetic knockouts, Purpose: to inactivate or knock out a specific gene. Mechanism: CRISPR introduces double-strand breaks (DSBs) at target sites. The cell's repair machinery often uses non-homologous end joining (NHEJ), which can result in insertion or deletion indels that disrupt the gene's function. 2. Gene knock-in. Purpose: to insert specific DNA sequences at a precise location. Mechanism: CRISPR creates a DSB, and the addition of a DNA template allows homology-directed repair (HDR) to insert the desired sequence. 3. Base editing. Purpose: to change a single DNA base, e.g., converting an AT pair to a GC pair. Mechanism: CRISPR is fused to a base editor like cytidine deaminase or adenine deaminase, which performs the base conversion without cutting the DNA. 4. Prime editing. Purpose: to make precise edits, including small insertion, deletion, and substitution without requiring DSBs or donor templates. Mechanism: a fusion of Cas9 nickers and a reverse transcriptase is guided by a prime editing guide RNA peg RNA to specify the edits. 5. Transcriptional regulation. Purpose: to activate or repress genes without altering the DNA sequence. Mechanism: CRISPR is used in a dead or catalytically inactive Cas9 form, dCas9, fused to transcriptional activator or repressor. 6. Epigenetic modification. Purpose: to modify epigenetic markers such as DNA methylation or histone acetylation without changing the DNA sequence. Mechanism: CRISPR dCas9 is fused to epigenetic modifier like DNA methyltransferases or histone acetyltransferases. 7. Chromosomal rearrangement. Purpose: to create large-scale chromosomal modification such as inversion, translocation, or deletion. Mechanism: CRISPR introduces breaks at multiple loci to induce rearrangements. 8. Gene tagging. Purpose: to add molecular tags, e.g., GFP or flag, to genes for tracking or functional studies. Mechanism: HDR is used to integrate the tagging sequence at a specific genomic site. 9. Gene activation. CRISPR-A. Purpose: to enhance the expression of a target gene. Mechanism: dCas9 is fused to transcriptional activator to upregulate gene expression. 10. Gene repression. CRISPR-I. Purpose: to suppress gene expression. Mechanism: dCas9 is fused to transcriptional reception such as KRAB to block transcription. How can this technology be used for patients with sickle cell diseases? CRISPR can potentially cure sickle cell disease by either correcting the genetic mutation in the HBB gene or by reactivating fetal hemoglobin production in red blood cells. The current method involves editing the patient's stem cells outside the body, then reinfusing them to produce healthy blood cells. This approach holds promise for long-term relief and possibly a permanent cure, but challenges like safety, delivery, and cost remains.
Lastly, we'll be looking at ethical concerns with the use of CRISPR. Lastly, The use of CRISPR, a powerful gene editing tool, has revolutionized biotechnology and medicine. However, its potential for modifying genetic material in living organisms raises several ethical concerns, some of which we'll discuss as follows. Firstly, there's germline editing and heritable changes. One of the most contentious aspects of CRISPR is its ability to edit germline cells such as sperm, eggs, or embryos. These changes can then be passed on to future generations. Secondly, there's equity and access. The potential for CRISPR to revolutionize healthcare by treating genetic diseases such as sickle cell anemia or cystic fibrosis is immense. However, access to this technology could be unequally distributed. Thirdly, there's environmental impact and ecological concerns. CRISPR is also being used to edit the genomes of plants and animals. Editing the genetic makeup of wild species or crops could have unforeseen consequences on biodiversity and the environment, particularly if a modified organism outcompetes native species or introduces new vulnerabilities. Lastly, there's bioterrorism and misuse. The ability to edit genes with relative ease could be exploited by individuals or groups with malicious intent to engineer harmful genetic modifications in humans, plants, or animals. Now, these are just some of the ethical concerns. There are more.
Thank you. Thank you.